Hi, this is going to be a series on protein peptide docking. We will begin with downloading protein using Protein Data Bank. Next, we will be preparing the downloaded protein for docking using Chimera X. Finally, docking will be performed using HeadDoc web server and the result will be visualized using PyMol. So let's begin with the identification of the target protein. So your protein should be something that plays a very important role in the survival of the target pathogen or in causing a disease. And this will require a thorough review of literature. And for the tutorial, I will be using MUR-E from S. aureus, which is an essential bacterial ligase enzyme involved in the biosynthesis of peptidoglycan. So now docking studies require 3D structure of both receptor as well as ligand. So for the 3D structure of our target protein, we will be using Protein Data Bank, which is basically a repository for 3D structural data of protein as well as nucleic acid. So now let's go to RCSP Protein Data Bank. This is the home page, and now let's search for our target protein MUR-E. As you can see now, multiple PDB structures appear, and you can also filter them based on the source organism of your interest. I am going to select Staphylococcus aureus, and now we are down to 2 PDB structures. So generally you will get more number of PDB structure, which can be quite overwhelming as to which structure should you choose for the molecular docking studies. So first, the structure should be experimentally validated and not predicted using in silico tools, and next you should look for resolution provided with the structure that are determined by X-ray crystallography or electron microscopy. So lower the resolution, better will be the structure, and resolution up to 2.5 is considered good. So I'll be selecting 4C12 based on the resolution, and on this page you will find all the details about the structure. So this 4C12 is known as the PDB ID, and you will find other details such as classification of the protein, organism from which it has been obtained, or if it contains any mutation, and the resolution of the structure, and the overall quality of the structure. Now further moving, you will find the literature, which is very important. It contains all the information related to the structure, its domain, any mutation, active site residues, etc. Now moving to the macromolecule section, you will find other details related to the structure, such as the number of chains it contains. So this one contain only chain A, but in many cases you will find multiple chain, and you need to check if the protein works as a monomer or a dimer or a tetramer through literature review, and keep only that chain and get rid of others, which I will be explaining in the part 2 of this series. So next you will find the sequence length, the number of amino acid it contains, and mutation, and since it's an enzyme, the EC number. Next you can find the UniProt ID, which we will be talking a little bit later. And in the small molecule section you will find all the ligands that are not protein but part of the structure. Now moving to the UniProt database. So the next important thing that you need to consider is if the structure is complete or not. So in this you will go to the structure section. So here you can see all the PDB structure listed along with the resolution as well as sequence length. Here for my protein, 2 PDB IDs are listed, and both of them contains complete sequence length. But in many cases you will see that the sequence is starting from 50 or 60 amino acid residues, and in that case you need to check that whether the sequence contains all the important residues or not. If it contains all the important residue, then the incomplete structure will also work for the molecular docking studies. Okay, so now going back to the PDB database, how to download the files. So you will be downloading the legacy PDB format for the 3D structure, and this will be downloaded in the .pdb format. All the 3D structure files will be downloaded in the .pdb format. The second file that you will be needing is the FASTA sequence file. So with this we come to an end on how to obtain 3D structure of the target protein from Protein Data Bank.